Ventilation, Chapter 122. Ventilation. In general, the word ventilation refers to circulation of replacement of air or gas in a space. In respiratory physiology, ventilation is the rate at which air enters or leaves the lungs. Ventilation in respiratory physiology is of two types: pulmonary ventilation, alveolar ventilation. Pulmonary ventilation. Definition. Pulmonary ventilation is defined as the volume of air moving in and out of respiratory tract in a given unit of time during quiet breathing. It is also called minute ventilation or respiratory minute volume, RMV. Pulmonary ventilation is a cyclic process, by which fresh air enters the lungs and an equal volume of air leaves the lungs. Normal value and calculation. Normal value of pulmonary ventilation is 6000 ml, 6L/minute. It is the product of tidal volume, TV, and the rate of respiration, RR. It is calculated by the formula, pulmonary ventilation equals tidal volume times respiratory rate equals 500 ml times 12/minute equals 6000 ml/minute. Alveolar ventilation. Definition. Alveolar ventilation is the amount of air utilized for gaseous exchange every minute. Alveolar ventilation is different from pulmonary ventilation. In pulmonary ventilation, 6L of air moves in and out of respiratory tract every minute. But the whole volume of air is not utilized for exchange of gases. Volume of air subjected for exchange of gases is the alveolar ventilation. Air trapped in the respiratory passage, dead space, does not take part in gaseous exchange. Normal value and calculation. Normal value of alveolar ventilation is 4200 ml, 4.2 L/minute. It is calculated by the formula, alveolar ventilation equals, tidal volume, dead space, x respiratory rate, equals, 500 to 150, ml times 12/minute, equals 4200 ml, 4.2 L/minute. Dead space. Definition. Dead space is defined as the part of the respiratory tract, where gaseous exchange does not take place. Air present in the dead space is called dead space air. Types of dead space. Dead space is of two types: anatomical dead space, physiological dead space. Anatomical dead space. Anatomical dead space extends from nose up to terminal bronchiole. It includes nose, pharynx, trachea, bronchi and branches of bronchi up to terminal bronchioles. These structures serve only as the passage for air movement. Gaseous exchange does not take place in these structures. Physiological dead space. Physiological dead space includes anatomical dead space plus two additional volumes. Additional volumes included in physiological dead space are, 1 air in the alveoli which are non-functioning. 2 1 and some respiratory diseases alveoli not functioning because of dysfunction or destruction of alveolar membrane. 3 air in the alveoli, which do not receive adequate blood flow. Gaseous exchange does not take place during inadequate blood supply. These two additional volumes are generally considered as wasted ventilation. Wasted ventilation and wasted air. Wasted ventilation is the volume of air that ventilates physiological dead space. Wasted air refers to air that is not utilized for gaseous exchange. Dead space air is generally considered as wasted air. Normal value of dead space. Volume of normal dead space is 150 ml. Under normal conditions, physiological dead space is equal to anatomical dead space. It is because all the alveoli are functioning and all the alveoli receive adequate blood flow in normal conditions. Physiological dead space increases during respiratory diseases, which affect the pulmonary blood flow or the alveoli. Measurement of dead space, nitrogen washout method. Dead space is measured by single breath nitrogen washout method. The subject respires normally for few minutes. Then, he takes a sudden inhalation of pure oxygen. Oxygen replaces the air in dead space, air passage, i.e. the dead space air contains only oxygen and it pushes the other gases into alveoli. Now, the subject exhales through a nitrogen meter. Nitrogen meter shows the concentration of nitrogen in expired air continuously. First portion of expired air comes from upper part of respiratory tract or air passage, which contains only oxygen. Next portion of expired air comes from the alveoli, which contains nitrogen. Now, the nitrogen meter shows the nitrogen concentration, which rises sharply and reaches the plateau soon. By using data obtained from nitrogen meter, a graph is plotted. From this graph, the dead space is calculated, Fig 122.1. The graph has two areas, area without nitrogen and area with nitrogen. Area of the graph is measured by a planimeter or by computer. Area without nitrogen indicates dead space air. It is calculated by the formula, ventilation-perfusion ratio. Definition: ventilation-perfusion ratio is the ratio of alveolar ventilation and the amount of blood that perfuse the alveoli. It is expressed as VA/Q, VA is alveolar ventilation. Q is blood flow, perfusion. Normal value and calculation. Normal value. Normal value of ventilation-perfusion ratio is about 0.84. Calculation. Alveolar ventilation is calculated by the formula, alveolar ventilation equals, tidal volume, dead space, times respiratory rate. Equals, 500 to 150, ml times 12/minute. Equals 4,200 ml/minute. Blood flow through alveoli, pulmonary blood flow. Equals 5,000 ml/minute. Therefore ventilation-perfusion ratio equals 4.200/5000 equals 0.84. Significance of ventilation-perfusion ratio. Ventilation-perfusion ratio signifies the gaseous exchange. It is affected if there is any change in alveolar ventilation or in blood flow. Ventilation without perfusion equals dead space. Perfusion without ventilation equals shunt, wasted air and wasted blood. Ventilation-perfusion ratio is not perfect because of existence of two factors on either side of alveolar membrane. These factors are, physiological dead space, which includes wasted air, see above. Physiological shunt, which includes wasted blood, Chapter 119. Variations in ventilation-perfusion ratio. Physiological variation. Ratio increases if ventilation increases without any change in blood flow. Ratio decreases if blood flow increases without any change in ventilation. In sitting position, there is reduction in blood flow in the upper part of the lungs, zone 1, then in the lower part, zone 3. Therefore, in zone 1 of lungs ventilation-perfusion ratio increases 3 times. At the same time, in zone 3 of the lungs, because of increased blood flow ventilation-perfusion ratio decreases, Chapter 119. Pathological variation. In chronic obstructive pulmonary diseases, COPD, ventilation is affected because of obstruction and destruction of alveolar membrane. So, ventilation-perfusion ratio reduces greatly.